Thank you. You know, you're starting to get old when people start saying you've been doing something for a long time, you know, but it's true. It's been a while, and it's a real pleasure to be here. First ISMB was over 25 years ago, and just want to really thank the organizers doing both NetBio and microbiome for inviting me. I do want to start in Canada. We're trying to do a lot of reconciliation with Indigenous peoples there who have, for millennia, been, taking care of their land, very sustainable interaction with the environment, and we're trying to learn a lot from them right now and integrating more holistic approaches into work. And so I'm sort of integrating a lot of holistic approaches actually into my research as well. And do want to note that I come from these unceded traditional territories of the Coast Salish people, including the Tsleil-Waututh, Coquitlam, Squamish, and Musqueam nations. And there's a picture of my land, the lands that I enjoy. So I have to say, I'm going to start with microbiome research just exploding right now. How many people here are you guys relatively like new to going to this microbiome or NetBio network? So they're like these sessions. Yeah, so there's quite a few new people here. So I'm going to try to make sure I'm inclusive of some of you that don't know so much and cover both areas. But I have to say, just look at the amount of, microbes are almost everywhere, and just they're being used in so many areas, from cancer to the bioeconomy to environmental monitoring. I would be remiss if I didn't include a cytoscape visualization. Nothing like those amazing pictures, and I love that artistic, that poetry was, poetic description was great too. But the microbiome research is exploding. We've got even on the International Space Station microbes being profiled. And of course, we know microbiome research has really arrived. There's now a game, just so you guys know, that you can get for free and play about, amaze your friends with giving them botulism. But basically, microbiomes have been increasingly investigated as sentinel predictors of health, including ecosystem health. This is just a little sampling just to give you a sense of all of the different kinds of metadata that's being collected essentially for these different microbiomes. And, there's everything from toxicology studies. I think I can use my mouse. No, it doesn't work. Okay. Yeah, okay. So from toxicology studies to looking at fecal contamination, detecting fecal contamination, to snow algae in the mountains as a predictor for climate change. And also one that really affected me was the beluga skin microbiome. Just so you know, belugas in the St. Lawrence estuary between Canada and the US, it was so polluted that when the animals would die, their bodies would be treated as toxic waste. Okay, so it has been transforming. They're really changing and cleaning things up, but because these are apex or top level predators, they get all these toxins concentrated in their bodies. And so they used to harpoon them to get little bits of their blubber to check them. And of course, the animals are very smart and they learn how to avoid the harpoons. Now what we can do is we can swab them. Isn't that nice? Swab them with and get their microbiome. And they don't mind that. So as a way, and it's used as sentinel for predicting their health. And it works, it's surprisingly well. So the also microbiomes and their roles in health, microbiome, gut microbiome can have impacts in everything from asthma, I'll talk about later, to impacting transplant rejection. And one of my favorites is doing analyses where you're looking at microbes that are naturally pleasant in environment, and how they can be used to help remediate some of the actions that we've taken. In this case, microbes naturally at a site, helping to transform copper at a proposed mining site to help clean it up. But all of this leads to this, what I call microbiome metadata madness. It's just insane, the amounts of different kinds of data being collected around microbiome data. I also want to say that's pretty well a picture of me trying to generate this with AI, this picture, because ChatGPT kept insisting on making a fish without a tail, and I was trying to tell it did not, there was no tail, and here's a picture of it with a tail then, and there was no tail, and I don't know, it made some sort of shark fish-like thing there. But anyways, the point I was trying to make is that there's all this different kinds of data around it. So I'll talk about microbiome research, but I am going to focus on this idea of getting data organized with a focus on quality and some of the issues that we've learned as we go forward, and with sort of more integrative analyses. So first, just for those that are new to this, I just have to put in a little plug for those. Just remember, you're always doing science, suitable experimental design, sequencing, pay attention to what sequencing method you're doing, get enough depth. If you're a bioinformaticist, talk early to people and tell the researchers that are doing microbiome analysis to make sure they are getting enough samples and depth of sequence, or it's going to be very hard to get anything. Make sure you're collecting those metadata samples, and pay attention to things like timing, the reference sequence database. In the coming talks, you'll see, I think, some discussion of like improving reference sequence databases, which is a big deal. Watching out for over predictions, allowing unknown predictions. We don't know everything, so if you watch out for incorrectly predicting, say, taxa A, because it isn't actually in your reference database, so it sort of says, oh, it's very similar to taxa B, and calls it taxa B. So we have to be very careful about how we're doing those analyses. I also just want to put in one brief caution about recovery of genomic islands and other mobile genetic elements. Genomic islands are sort of clusters of genes of potential horizontal origin, and antimicrobial resistance genes. Metagenome assembled genomes can be good, but you have to be very careful with them. So we did one simulation where you're sort of simulating this, the metagenome assembled genomes, where you're assembling these reads. And the chromosomes were recovered well, but the plasmids, less than 30% were binned correctly. AMR genes, less than 45% were binned correctly. And in fact, of the correctly binned AMR genes, or antimicrobial resistance genes, which we care a lot about in many microbial studies, 0% were plasmid located, even though they are certainly, a lot of them are on plasmids. So the main point is that you can use a combination of MAGs and read-based technologies, or use longer read technology, including with MAGs and longer read technology. But just be aware that there is this challenge with the, with MAGs, and pay attention to what you're trying to identify and make sure you use the right methods for it. Also, genomic islands aren't very well identified in short read draft genomes as well, just for reference. But I also want to do a shout out to these critical assessment groups like CAMI, that's going to be talked about later, critical assessment of metagenome interpretation, CAFA, all these different groups. It is really important in this age of AI is just causing just a transformative storm of information. And we have a problem with we need to make sure that these critical assessments and this accuracy is still front and center, or it's going to get very messy very fast. So I just really want to encourage, I think we all need to step up and make sure it's clear to funders, for example, that this kind of work is absolutely critical. CASP, for example, has been under threat, and others too. So I also just want to mention about getting data organized. We are initiating this little project that's iMicroSeq. It's a data portal that builds on another thing we made, this VirusSeq data portal with metadata specifications. And I'm not going to go into this at all. This is just starting up, but I just wanted to bring up a point just in case. I would love to see if people have comments about this, but our funders and some other researchers said they feel like for when we do public communication about microbial monitoring, that we should just use also the term eDNA, which tends to be traditionally used for animal DNA barcoding, and that we should just put everything together as eDNA is basically environmental DNA, even though we look at COVID, we're monitoring RNA, but anyways, we should just call it all eDNA to be inclusive in animal and microbial monitoring, and I would love thoughts on that. This project, we are trying to put things together. We are trying to take the DNA barcoding and the metagenomics and the amplicon-based microbiome profiling and trying to do metadata specifications that are interoperable. You never can have one standard, but you can have scripts and ways to move things between different standards and have interoperability frameworks. So that's what we're sort of working on here. And if you're interested in that, please let me know. I do want to do also another shout out to Emma Griffiths, my co-lead with this iMicroSeq project, where we're they basically developing these contextual data specifications. And this is for like wastewater, like taking the Public Health Alliance for Genomic Epidemiology, which was sort of microbial genomes. This is being implemented in NCBI, and the taking those data specifications and expanding it to handle wastewater. Now we're expanding it to handle water. And I just wanted to bring up that this is again about just trying to come up with some modular ontology based, that's controlled vocabulary with hierarchy, ISO compatible standards to try to make it that we can compare things across these different domains as we try to get more holistic in our analyses. But I'm going to spend the rest of the time, just checking my time here, talking about magnificent metadata madness, or my God, it's a lot of stuff. But basically, this is integration of microbiome data with other diverse data, and just showing how we found it's useful in driving some innovation, but some of the points that we think are important to keep in mind. And I just wanted to acknowledge Jeff Windsor, shown there on the left, and others in our group who've been really leading this work, and really privileged to have such great colleagues. So this is based on, this example is based on this Canadian child cohort study where we integrated, we've got now 73 million different data points. This is an insanely diverse data set of information on 3,400 roughly child participants that were born healthy and being tracked longitudinally over time. It's got questionnaire information, clinical information, all the environmental information by postal code, sample derived data, everything from how many hours a night you leave your windows open to mental health information, pollution information. For the omics data, there's chemical analytics, epigenetic data, genetic data, and microbiome data. And the microbiome data includes gut data, breast milk microbiome, nasal microbiome. Certainly, one challenge is we haven't completed all of the things for all of the people. So we've got some, or sometimes there's subsets of people that have been, but this is data that you can put in. We try to control it to make sure that it doesn't get too away, not in terms of for publication purposes, but just to protect the participants in terms of how analyses are done, identifiability, that kind of thing. So you have to put in sort of a concept proposal if you want to use the data, but certainly this is available. And I wanted to also make you aware that what we're increasingly doing is looking at sort of exposome, as we call it, mostly because I think somebody said, oh, you got to do your next paper, you got to refer to that term for some reason. But exposome and microbiome analyses for asthma. Most of you in this room do not have asthma, but it is the most chronic common disease in, or common chronic disease in children is disproportionately impacts the healthcare system. If we look at that child cohort and we see who are the kids that have highest unscheduled doctor's visits and highest medication use, all the correlating variables are all things to do with asthma. They really use the healthcare system a lot. So we're really interested in what causes asthma, how can we prevent it from occurring in the first place. There was a talk earlier in NetBio that was bringing up really cool analyses of, looking at contributions of genetic versus environmental components, and mentioning how asthma has both a genetic component and an environmental component, very significantly for both. And I just want to mention that microbiome is thought to play a role. So there's a lot of interest in the microbiome in this context, with multiple asthma phenotypes being described, and I'll come back to that later. One of the thoughts is this quote old friends hypothesis, or the idea of exposure to good microbes that we've been exposed to for, hundreds of years in our ancestors, that those were used to help appropriately stimulate the immune system, and that asthma is basically an immune disorder caused by a lack of exposure to these good microbes. But the thing is, how does that normal microbe exposure get disrupted? If that's the case, we wanted to just use an agnostic, let's look at all the exposures and see what exposures in this rich data set are associated with asthma. So a year ago, I presented the next 3 slides in a talk. So I'm going to go through them pretty quickly, but some of you won't have seen that. But basically, we did an exposome-wide association study for asthma, looking at doing logistic regression for 2,705 variables prenatal to 5-year exposures, including clinical data, and a bunch of covariates, and then did a more in-depth XWAS with more covariates, and corrected for multiple testing, of course. And in short, this is one I showed before, that there's definitely some significant positive and negative associations with 5-year asthma. And one of the surprises we found was prenatal disinfectant cleaner use potentially impacting the child. We tend to think of it as, the kid's getting an antibiotic or getting some sort of disruption in their microbiome directly, not that the mother would prenatally be using cleaning products, and that would result in 5 years later that child getting asthma. So that was very surprising. And even if they had cleaning product exposure, like for example, they're house cleaners. So some of these people that have the highest risk of their child developing asthma, or they're using weekly oven cleaner. And we're like, who is using weekly oven cleaner, right? That's crazy. But it turns out to be that it's like house cleaners and people like that will be using cleaning products a lot. And they have a much higher risk of their child getting asthma, even if they stopped using the cleaning product at birth, they go on a maternity leave. So they use the cleaning product prenatally, stop it, still increased risk of asthma. So because of all this diverse data, we're able to look at things further, and found that there's actually epigenetic changes occurring detected in the cord blood at birth of in inflammatory genes for those who have had used these sort of excessive prenatal cleaners and disinfectant uses, like daily hand sanitizer, very significant, pretty significant cleaning product use. But now to move to new stuff is, really what we wanted to do though is to really say, okay, so we've got that, but we've got these other variables. Can we try to get at sort of more causation? Because of course, correlation does not imply causation, right? And so do these variables actually correlate with each other? Is it just that it's one thing and then they just all correlate with these other things? But actually, when we look at it, we see that there's actually pretty limited between category correlations, and limited sort of mechanistic relationships. So for example, I don't know how easy it is to see this, but, and where there says antibiotics there's, number of antibiotic courses taken from birth to 12 months doesn't correlate with any of these other variables at any appreciable level. And some of these correlations are like 0.1 something, so don't really take it too seriously. However, this was really useful to be able to see that, but also, we could correct for confounders. We could say, well, but let's just take, some of these guys and let's correct for confounders, watching out for colliders, but correcting for confounders, saying, is antibiotics still significant? And yes, it is still significant when you correct for some of these other ones, cleaning products as well. There's another one here, phthalate levels that also, this is like found in plastics, if you have high phthalate levels in the dust. But do note that it does correlate weakly with this other variable that's associated with asthma, which is vitamin D supplementation. Children living in northern climates, their families are encouraged them to give vitamin D supplements if they're breastfeeding, because they don't get enough vitamin D because of the lack of sun exposure. I personally, with my kids, I just got them out in the sun and just like, in winter, just give them something. But basically, vitamin D supplementation does correlate with the dust phthalates, which also correlates with the oven cleaner use. So there is some possible weak relationships there that need to be explored. But the antibiotics, for example, is not correlating with any of them, implying again that there might be multiple things, and one is antibiotic use after birth and one is prenatal. So it's pretty likely that they're not totally related. I do want to mention there's reverse causation that can occur. And so one point I wanted to make was it was beneficial looking at many different like this network of metadata and these correlations and doing a bit of an analysis. That was helpful. Also, just looking out for things like reverse causation. So the medication of using albuterol shows up as very associated with asthma. Albuterol is a medication given to asthma patients. So it's just that they are getting that medication because they had asthma, and so therefore it's associated. One of the worries I had was, well, antibiotic shows up in the same way. Is it actually just that these kids getting asthma, they have lung inflammation, they're more susceptible to infections? Do they just end up getting more infections? They get more antibiotics, and it's actually all about the prenatal cleaning products. It's a big deal, causing asthma, and then the asthma makes them more susceptible to take a bunch of antibiotics. But I want to bring up my second point. In addition to having all that rich network of data, it was also very helpful to have like population-based natural experiments. And I encourage, with microbiome research and even network-based analyses, we do have to pay attention to different kinds of experimentation, like lab-based stuff, cohort-based stuff, population-based data. They can all sort of have their benefits and disadvantages. And in this case, we had a really handy policy change in British Columbia, Canada. It's a population of 4.7 million people at the time of the study. Actually, we're at 5.7 million. There's a lot of people coming to BC right now. But this was a study, I guess, looking at, 10 years ago. But we've had a big increase. But what they did is they reduced antibiotic prescribing. They said, and I encourage you to do this, if you have a kid and they get sick and you're like, oh, they might have a bacterial infection, they should get an antibiotic. You go to the doctor, they would have a policy of, say, just hold off I'll give you the prescription, but just wait 2 days and see if it gets better or worse. If it gets worse or stays the same, take the antibiotic. If it doesn't, it starts to get better, just don't take the antibiotic. I did that with my own kids, and twice I had a prescription in hand, waited 2 days, it got better. And I'm like, oh, geez, I would have thought that was the antibiotic making it better, right? But it just got, naturally, you tend to go when you're at the peak of an infection with kids, and sort of, in short, they're pretty resilient. And this reduced antibiotic prescribing policy did result in reduced use of antibiotics and a corresponding reduced asthma incidence, including after excluding children who received antibiotics for respiratory symptoms. And also note there's a decreased gut microbiome alpha diversity at 1 year associated with this antibiotic use. along with selected changes in taxa. So this to me tells me that, and you can see this paper here, it's got more information about this, but the main point is that population-based data combined with this cohort data, certainly, and some animal model arguably data, does suggest that antibiotics really do have a role to play in disrupting the microbiome that can lead to asthma. But we still really don't have this worked out exactly how that's occurring, right? So I wanted to check too, what about other asthma associated variables? Do they impact the microbiome? One of the other issues I want to bring up when you're doing microbiome analyses is that the timing of microbiome sampling really matters. And it's not necessarily at the time of exposure. So we have an issue with, you want to look at this microbiome, but then it wasn't right when they took an antibiotic. One exception we have with this work is that the vitamin D supplementation tended to be given to a child more likely on the day the gut microbiome was sampled, because they would just do this daily supplementation. So then you get this gut microbiome sampled at age 3 months, and we tried to make it like just a range of time between 2.5 and 4 month samples. And in short, we do see a reduction in diversity, and exclusively breastfed infants have a change in their alpha diversity when they have take vitamin D. Interestingly, with the not breastfed children, which is shown at the bottom there, you can see these DVI sal drops are basically vitamin D. And we did a whole analysis to look at vitamin supplements that babies take, and it's basically 95% vitamin D that they're taking. But so we don't see a change in diversity, including after controlling for confounders, but we do see this lower species richness and evenness in breastfed infants. I want you to be aware that already breastfed infants do have lower diversity, but that's not necessarily a bad thing. There's lots of good microbes there. There's no association with breastfeeding and asthma being increased. If anything, it's protective, but it seems to be like this extra drop down is associated with an increased risk of asthma. There's also taxa that we could look at and see the 3 month gut microbiome. Again, we tried to sort of be careful, like processing time was less than 96 hours. It wasn't like that sampled and then sequenced much later. But this is, with Maslin 3, looking at vitamin D and controlling for these different confounders. And in short, we do see some increased abundance and prevalence of some Bacteroides and decreased abundance or prevalence of some Clostridium, Strep, and Bifidobacterium. I have in brackets the species names because I get very nervous about, I don't know, some of these situations of how accurately we're really detecting species level. But I'm going to finish up now and say that I can't bring up all the analyses we've done and all the data we've done, but what I want to get across is there's this just absolutely mad amount of data that you can look at, but it's very powerful, and I'm really enjoying it, being able to tease out what's going on. And in short, definitely antibiotic use seems to be notable and playing probably a role in mediating causing asthma. Vitamin D is associated. When we start correcting for other asthma-associated things, we don't see the vitamin D. We still see the vitamin D showing up, but the serum cytokines are not changed. So I'm sort of a little suspicious. I don't think we should go around saying that vitamin D's supplementation is a problem for breastfed infants right now. There's prenatal cleaners. Definitely, there seems to be something going on there, including an association with epigenetic changes in the cord blood. Phthalates, I haven't gotten into, but we do not see a gut microbiome change there. And in prenatal cleaners, there's a timing issue of we haven't sampled prenatally the microbiome in the mom. We would love to look at the skin microbiome, but The other thing I didn't get a chance to talk about is adrenic acid, this breast milk metabolite that seems to be more a marker of certain inflammatory conditions and a sex-dependent association with females who tend to get more prevalent asthma later. But the main message I wanted to emphasize here is that it does seem like there's multiple causes. This makes sense because clinicians have noticed different phenotypes of asthma, and that this sort of, but getting this microbiome data together with this other metadata is very powerful. So cautionary notes, just 2 more slides if I can. We need to iteratively study with machine learning with a careful selection of variables. You can just stick a bunch of variables together, and we do find we have to sort of try all in and then try subsets where we're picking certain ones to try to more robustly tease out, which are the factors that are really playing a role. And I think it is important to complement machine learning methods with other methods like these XWAS, these logistic regressions, because the logistic regressions, the pairwise logistic regressions we did, there's no imputation there. That's just all data, real data. With machine learning methods, which we've also used, and also show phthalates and cleaning products showing up, antibiotics showing up as being in the model for predicting asthma. They are really identifying the more common factors in general. So there is a need for being really careful about using different kinds of approaches. More curation, this data organization is needed. I don't have time to go into things like tools for more interactively looking at correlation networks, but there's been some nice tools that have been developed, but I do think tools to allow people to do very local based analyses are great. And just a real shout out to the Cytoscape developments have been fantastic. Defining healthy in children is the one last thing I want to leave you with is we started looking at low unscheduled doctor's visits and high unscheduled doctor's visits. And we noticed in the data, it looks like it's due to like, you can be low unscheduled doctor's visits because you're healthy, but also because you have low healthcare access. You can have high unscheduled doctor's visits because you're unwell, but also because you have really anal parents who are taking you to the doctor a lot, okay? Seriously, that's what it looks like. And so there is an issue here with really needing to be careful with this metadata as we try to look at what is a healthy microbiome, what is a healthy child, which is one of the things we're focused on. Again, accurate, well-organized data is important. Correlation analysis of the rich metadata as a network can be very useful. Complement with sort of population level, natural, like we need to get out there in nature and be looking at measurements in nature. Watch for the timing of microbiome measures versus some sort of exposure or condition. And with the example study of asthma, we definitely have to appreciate that there is probably a gut microbiome mediated cause that involves multiple causes. And that I want to note that this can really lead to community level action. We can have policy change on prenatal use of cleaning products during pregnancy, right? And expanded antibiotic stewardship policies to help really reduce that healthcare burden. So I will end with just saying, but I love this microbial metadata madness. It is awesome. I just want to get along with these guys and thank the great piles of people I've been had the privilege to work with, including students, postdocs, so many people that have been absolutely essential. And thank you. And last plug for MicroBioInfo Slack. It's in the bottom right corner there. We don't tend to publicize the link because we don't want it to be insane. But if you're interested in joining, it's a fantastic Slack network of people doing microbial bioinformatics. Just contact me, or you can email me or whatever, and I'll send you the link to join that Slack. Thank you so much. Happy to take questions.
Yeah, thank you very much. We have time for a couple of questions. Are there any questions?
There's somebody over there.
One. So there are actually microphones.
Okay, thank you.
So I assume that in your model you use the microbial composition that you measure in these children or their mothers. So I'm wondering how reliable is this microbial composition measured at a single time point? How important would it be to have longitudinal data looking at multiple time series of mother and child? Because just measuring one maybe, I don't know.
Yeah, absolutely.
Comment on that.
Yeah, sorry, you said, I missed one word though. You said you're wondering about when I did the microbial analysis at,
Composition, yeah.
yeah, but then you said something and I missed the word.
I guess you use that in your model.
Oh, yeah, okay, yeah, so,
So how do you
um,
deal with the fact that it's a single time point? Or
yeah, so a couple of things. We did for the exposome kind of analysis, that was not including the microbial data. We're sort of separately looking at the microbial data, so it's not part of that model of exposures. We're sort of looking at what those exposures are and then seeing what is showing up in terms of microbial changes associated with those exposures. But absolutely, we're interested, of course, in incorporating these. We do have multiple microbial data. I can go back to, if I can, to the beginning. But I do agree that doing time courses, this is about sort of, so we've got 3 months in 1 year, for example, for the gut microbiome. There's also skin microbiome that we haven't looked at. One of the problems we have right now is that we don't have a gut microbiome of the mother. We don't have a skin microbiome of the mother. So that's a concern because they obviously probably with this cleaning product use would have presumably some impact on it. So I really think this just leads to new experiments that need to be done that are properly organized. But does that really, in short, yes. We need to do more longitudinal analyses. You have to be very careful about, when you don't see something and you're not measuring it at the right time, you might not be capturing it. But one thing I did like about the vitamin D is that generally these people are taking it daily. So you're sort of getting that experiment of the actual vitamin D use versus not. It isn't that we didn't confirm, like we didn't ask them though, did you give your kid vitamin D that day of the microbiome sample, which is a regret, but so we don't have that for sure. So there's noise, of course, in that data.
Okay, that's one more question.
Hi, I'm also from Canada. I'm actually part of Human Blood group. So I'm quite familiar with this, cohort study. And you talked about using these features or using these features for machine learning. And of course, you have multiple time points from prenatal to birth and such. What strategies are you thinking of to actually use them, considering the different time points of the microbiome or other exposure data in your machine learning?
Yeah, no, that's a great question. So we've been focused on trying to use the early, I didn't show the data, but too bad I didn't know. I should have done how you always add extra slides to a talk and then you never use them. So I was like, I'm not going to bother. And now I need to use it. But anyways, so we have machine learning we did with this earliest time point data, right? So within the first 3 months, and trying to use that to predict asthma at age 5 and age 8 to sort of try to err on the side of seeing what can we find early to predict, that trajectory. So we haven't really dealt with the issue of that longitudinal nature of it. We're sort of just trying to do early, but I do agree though that it'll be one thing I'm really keen on is that trajectories, right? We want to look at the kids. So for healthy kids, we want to look at kids will start with a bad start at life and they end up healthy, or they start healthy and they continue healthy, we want to know both of those scenarios, right? And we also want to know the kids that start healthy and go really bad, you know, what's happening with them. So we're looking at those factors, but it's tricky because that correlation does not imply causation, right? I can't emphasize it enough. Yeah, sorry, did you have a?
Okay, one last
Yeah.
quick question and
Yeah,
quick answer.
it's a-
Can I change it?
Great talk. I wondered about you mentioned confounding factors, and you also mentioned vitamin D being given by more by parents where there's less sun. I'm wondering about kind of the geographic confounders. It's a Canadian cohort, so I imagine that there's probably some geographic boundaries on that. But did you look at, latitude, for instance?
Yeah, that's a great point. So we did include, there's this really great site called Canue, CANUE.ca, and it's got all this environmental data, socioeconomic data. We incorporated that in. So we can find some things to do with like green space and blue space and that kind of thing. And in short, we don't see that, but I have to say the cities are all like, you know, we're like, it's a pretty similar latitude, but I have to say that's a very good point. And I think that there is definitely to do a lot of, there's differences in Vancouver versus Toronto versus Winnipeg in terms that we have to correct for study site. Absolutely.
Yeah, maybe if they're all of them about the same latitude, it's kind of control anyway.
That's a good point. Yeah. Yeah.
Okay,
Yeah,
we have to
thank
move
you.
on. Thank
Yeah,
you very
thank
much
you.
again.
Thank you.